Okay, hi everyone. My name is Rachel St. Clair. I'm a graduate student here at FAU. I work in the Machine Perception and Cognitive Robotics Lab under Dr. Barinholtz and Dr. Hahn. And Dr. Pavlovic has asked me to come and give part of this guest lecture today on machine learning for protein analysis. And hopefully we're going to integrate some of what you guys learned from Dr. Barinholtz previously, and we're going to actually apply it and do a bit of a hands-on tutorial, and then you'll be well prepared to listen to Dr. Hahn's lecture on advanced models. So before we get started, I included a picture of myself so you guys can see what I look like, so you don't think you're just getting a lecture from a computer or something. Okay, so let's get started. I don't have that many slides. We're going to mostly focus on the actual work in doing the tutorial. But first, I just want to give you guys an overview of what we're going to learn today. So, getting started with computational analysis. This is what we're going to go over today. There's tons of other stuff that's involved in this whole process. Especially the coding and stuff like that. So if you're not familiar with programming, don't worry about that. We're not trying to teach you every single detail of how to do something. It's just the overall process that's important. So if you wanted to go and tackle a problem on your own, you know the steps which you need to take to find the information you need in order to do the work you want to do. So let's get started with getting our data. First of all, we have this thing called Google Collaboratory Notebook. It is a free software provided by Google, if you can call it a software really. It's kind of an application. So if you click on this link, you can pull up the notebook that we'll be working in. I'll show you guys how to do that a little bit later. I just wanted to get everyone familiar. And if you want to go ahead and pull that up to follow along, you can go ahead. So let's talk about getting data. We want to get a lot of proteins to analyze, right? So one of the places we can do that, which we're going to see today, is called uniprot.org. And if we go to that website, it looks something like this. You guys have probably heard of it. And if you go to the manually annotated and reviewed section, you have all these different proteins. So the way these are stored, if you click on one of these, you get information about them. And more importantly, if you go up here to Format and FASTA, you can get the FASTA sequence. So hopefully everyone is very familiar with this type of writing of a protein, right? That's what we're showing here. We're using these one-letter codes to represent each of these residues. So that's what we're going to work with today. So another place that you can get data is from the Protein Data Bank Warehouse, which is this RCSB link. Feel free to check it out in your own time, as well as this PubChem. That's for chemical information, so for chemical sequences and chemical molecules, you're going to want to go to that link. All three of these have easily integratable applications with the stuff we're going to do today, so if there's a problem you're interested in, keep those in mind. So after you get your data, what do you do with it, right? In biology, in the real world, proteins live as something like this first image up here, right? They kind of look like, well, this is an antibody, so it kind of takes on this tertiary structure. And biology really prefers this type of format. However, for us as humans, this is not easily interpretable for me and you to say, hey, oh look, what's that residue right there, right? So that's why we write them in this FASTA format. However, this FASTA format, these letters, computers don't really like this either, right? So then we have to turn it into some computer format that the computer can read. And I know this probably sounds really obvious, but sometimes this is a step that takes the longest. It can be the hardest for students and people in general. So I'm going to show you exactly how to do that. It's really simple once you get the hang of it. You just need to keep things organized. So the image we have here at the bottom is an example of a one-hot encoding. So basically you convert each of these letters to a number, and then those numbers are going to be represented by the columns. And the rows is going to be the position of the protein. So this 0 corresponds with this M up here, position 0, right? So whatever M is, let's say we say M is going to be represented by the letter 2, it's going to get a little mark right here. So in the 0 position, we have M, which we're also calling the letter 2, or the number 2, and that's going to go here, right? So you can see how you would fill out the rows or positions on the columns are which letter it is, which is going to be represented by a number. This will make more sense later when we see it. So I just explained 2 ways for you to represent these strings. You can just turn these into a string of numbers, or you can represent it as an image. And this is very important for depending on which type of model you want to use. Okay, so let's talk about how do you organize your data. You're going to need a training set, a validation set, and a testing set. A training set is what you're going to, train on. That's when you're going to learn your parameters, right? That's when you're going to teach your model whatever it's supposed to do using that data. And that data is almost going to get memorized. So that's why you need a validation set. That's when you tweak your model, whatever your model is, and then you see, okay, I'm using the same training data, but when I use my validation, that doesn't change the model. It's just when I change configurations of the model, I want to see, does this configuration perform better or worse? So that's all having to do with setting up the model. And once you're sure that you have something that you like and it's working and it's good for your needs, then you need to check, does it generalize? So it's kind of like if I gave you a bunch of words in Japanese to memorize, and you memorize them, and then I gave you a new word you've never seen before, you probably wouldn't do very well at guessing what that new word means. That's overfitting, and Dr. Barinholtz probably talked about that. So that's why you use a test set. So you can see, okay, I think I have a good model. How good is it really when it sees data that it's never even heard of? All right, so that's data. So once you get it, you're going to want to do some basic stuff, and don't skip this step. I see it all the time. I've been a victim of it. You skip the basics, and then you end up way down the line having to redo a bunch of stuff because you skipped this step. So the first thing you want to do is get to know your data through and through. You want to know everything about it. You want to spend a lot of time on this section getting to know it. So for us, when we read in proteins, one of the big concerns is in our classes or 2 different groups of proteins that we're looking at, are the lengths similar? So we're going to look at that. We're also going to look at beginning and ending residues. So that could be like a big predictor of your model. You know, if one group always starts with one letter and the other one starts with a different letter, well, that's a dead giveaway. We want to make this a little bit tough to simulate a real world problem. The other thing you want to do in this step is discard outliers, obvious outliers. So here on the right, we'll see this plot later, but we're actually going to generate this plot, but it is actually a plot of 2 different groups of proteins and their respective lengths. So you can see that most proteins are under 200 amino acids long. So on the x-axis we have length, and on the y-axis we have the number of proteins in each class with that length. And very few are over 1,000. So let's say we had like very tiny amount there that were way out on 5,000, we would want to get rid of those. And we'll talk more about that later. Okay, so principal component analysis is a really easy tool that a lot of people do first, and it can actually give you insight into your data and actually be a predictive mechanism if you do it correctly. And if your data is less complex, then it needs to be evaluated by more computational analysis. So basically, principal component analysis, you've probably heard of it. It's a dimensionality reduction technique. So what that means is, let's say you have a protein that is 200 amino acids long, and there's 27 possible residues that can be in each position. Well, you can see that's kind of going to be a combinatorial explosion problem, right? If you try to brute force guess every single one in there, it becomes a factorial problem. So that's not going to be good. So what you want to do, and also for humans to visualize, you want to reduce that number of dimensions you have to search through to see which dimensions are the most important for separating the data. That's what principal component analysis is going to do. So you have two groups of proteins here, B and M, and you're going to use PCA, and you're going to try to say, okay, in dimension 1 and dimension 2, you can separate the data based on these things. Then you can go back and try to figure out what exactly is dimension 2. Is that position 37, position 100, something like that? You can kind of use your imagination to think what's going on here. And you see you get these nice clustering of groups. That doesn't always happen. That only happens when your data can be reduced easily. And we'll see later that may not be possible in some instances. So after you've done that, and maybe you've said, okay, well, I don't really know that much information about my data from PCA, I need to try something else. The next thing you should try is logistic or linear regression. And hopefully Dr. Bernholtz said a thing or two about this. But basically, again, we have two groups of protein, the blue circles and the red X's. And we're going to say, how do we define classes based on this? Obviously, it's really easy looking at this image because you only have 2 dimensions. But remember, for us and for most problems, there's going to be a lot more than 2 dimensions, and it's not possible to graph things really above 4 dimensions easily for us to understand as humans. So that's where logistic regression comes in. It's basically, the computer's basically going to build this probabilistic linear mathematical model to create this decision boundary that you see on the right. And it's going to happen in more dimensions than just 2. So then you can use this logistic regression model to predict unseen proteins. So that's that test set we were talking about earlier. So you'd use your train set, you could change your configurations with your validation set, and then you could get your test set, which is maybe a synthetic compound you came up with or something like that. Give it to your model and it says, hey, this is gonna belong to this class or whatever. Another cool thing about logistic regression that I'm not going to show you today, but it's important to keep in mind, is the coefficients imply feature importance. So when we talk about a linear mathematical model, what we're talking about is not just a straight line, right? This can be any sort of algebraic expression that mathematically would be determined linear. So what that looks like is, a times x squared plus b times x to the cube, blah, 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 things like that. So those a's and b's I just mentioned, those are your coefficients. And if you look at those, each of those coefficients will show you how important that particular feature is, x being your features. Don't worry about the details, just know that that exists. So later on when we're doing this stuff, you can kind of think of what would be a really cool application for you to do. So we're almost done, guys, with the slides, and then we're going to get to the tutorial. We want to talk about convolutional neural networks. This is the cool stuff, right? This is what you hear about in a lot of papers nowadays. It's all over Nature. It's all over the internet, basic media. You probably are familiar with a lot of the stuff I've said already, and you're probably familiar a little bit with convolutional neural networks. Hopefully Dr. Bernholtz explained all about these, and this is just a refresher. But basically, you're going to predict a class. You're going to predict whether or not something belongs to a group of proteins, in our case, with nonlinear models. So this is very different from logistic regression because we're using nonlinear applications and nonlinear mathematics. And it has a lot to do with advanced calculus and derivatives. It's not too hard once you get into the meat of it, But in order to execute these things, you really don't have to know any math. So I'm going to show you that that's true later on today. And what we're going to do is we're going to learn these feature maps, which is the second thing next to this bird in the image. And those feature maps is basically what the convolutional neural network is going to use to make its decision. And in this case, it's taking in an image and it's saying, is this a bird, a sunset, a dog, or a cat? And maybe it's going to say, oh, I see feathers. I have a feature map for feathers. I know that that is a bird. So we're going to do that for proteins. It's going to say, oh, I see, residue M in position 32. I know that is an antibody, or whatever it's going to do. These are really great for big data. You can also use them for smaller data sets. And when we say big data, we're talking about like millions of proteins. Small data sets would be in the thousands. So you can see that this already far exceeds most high throughput screening wet lab approaches, and it's why it's being used to aid researchers in their prediction pipelines. So as a wet lab researcher, you might need to determine something and create some synthetic compound, but instead of just jumping in with not too much background knowledge, you may want to run some computational models first so you can guide your decision-making process as a wet lab researcher. Alright, so now that you have all these great models, you're not done yet, right? So everyone thinks, oh, I made a model, that's great, that's awesome, and it is good, but you need to interpret your model, and the community of deep learning and AI is really putting out a call for this because a lot of people are saying that these models are black boxes. And while some of that is true, there is a lot of ways to interpret any type of model, and it's a lot of work. It's kind of like the pre-processing data gathering step. You have to spend a lot of time on it, and it might not necessarily be the fun part, but it's really important for understanding the entire process and what's going on. How is your model making its decisions? So I'm going to show you some very basic metrics to determine how good your model is, called sensitivity or recall, specificity, precision, area under the curve, and ROCs. And we're also going to visualize some feature maps. Visualizing feature maps is one way to look inside of a CNN and see how it's making the decisions that it's making. Sorry, I put a link down here that's really good on this sensitivity, specificity, and precision stuff. I've read this article probably like 10 times. It's very helpful if you ever get confused about this kind of stuff. So, last slide. What did we learn? This is the process. You have to formulate a worthy question to investigate. You have to get your data, format it, and clean it up. You have to try some more simple approaches before you get to the really fun stuff. You want to do basic statistics and early on machine learning to make sure that your problem can't be tackled with smaller, less computationally extensive models. Then, if those aren't enough, you need to decide on an appropriate model for your data in your question. This is very different from just taking someone else's stuff and applying to your own. You may need to make some serious modifications in order for it to work and make sense on your stuff. This decision point should be a lengthy amount of your time in this process. You shouldn't just arbitrarily choose. You should have some basic reasons on why you choose the model for you, and you can kind of get to know more about that decision-making process as you read more articles and if you choose to work on this kind of stuff. So then you build your model and you make sure it works. What I mean by that is you probably want to use a toy data set, something like cats versus dogs, or something very easy, just to make sure that your model is working. And you don't want to go through all the trouble of putting all of your special data into it and then getting really confused. I'm speaking from experience here, so just make sure you're taking the care and time to put the thought into each one of these steps before you rush on to the fun stuff, which is step number 7, train the model and test your data. It's the funnest thing ever. I love when I get to that step. And it doesn't last long, but it's great. Because that's when you really realize that you've done something and you have something useful that you can give to the world. And in order to give it to the world, you have to interpret your model properly and report your results. So that's the process. I hope you don't have too many questions. If you do, hopefully they will get cleared up here when we go ahead to the collaboratory notebook. Okay, so you click on this link. It takes you to this web page. I made this notebook. It'll probably say, hey, someone else made this notebook. Are you sure you want to use it? You can hit yes if you trust me, which you should. So now what you want to do is you want to go to File. You want to go to Save a Copy in Drive. And that is automatically going to make your very own copy, which you can add notes to, you can play around with, you can design your own models on top of this code. This is yours. I can't see it. It's totally just for you. So I'm going to go back to the original copy because I'm going to work off that. And first thing we're going to do, again, don't get scared by all the code. We're not trying to understand each line of code. We're just trying to understand the general process of how you would do a project from start to finish. And we're going to leave out a lot of details, but the goal is to get the basic overview down. So, another thing you want to do for later, you want to go up to runtime, and you want to go change runtime type. And it's going to say, hey, do you want to use a hardware accelerator? And you want to say, yeah, I want to use a graphic processing unit, a GPU. So you click on GPU and you hit save. And it's going to restart the kernel. So basically, Collab Notebook is just a way to use really expensive computers that Google owns for free. So you get to run your code on the internet with really expensive computers, and it's great. It's awesome. So we installed this stuff that we need to use, and now we're going to get some data from Uniprot. One more thing before we jump right in, I want to show you guys how to, if you're not familiar with coding, how do you navigate this. So if you want to add a text box, you go up here to text and you hit that, and now you can type some notes with the text box, right? And then you can just hit Shift Enter to get out of it, or you can just click out of it, right? You can just click out of it. All right, so maybe you don't want to do all that when you're taking notes because I'm talking really fast, and I do apologize. We just have a lot to get through. The other thing you can do is you can be in the coding cell. This is a coding cell. You can tell by the numbers on the side. You can just put a little hashtag, and you can just say type notes with hashtag or whatever your note wants to be. That way, when you run the cell, it won't become code. So do remember that if you put it on a new line, it's no longer part of the hashtag. It has to get its own hashtag. Okay, that's just the basics. Hopefully everyone knows that kind of stuff. If you don't worry. So we have a little function here that is going to go to Uniprot, that website we were at earlier. It's going to pull proteins of a specific number. So we want 2,000 proteins, 3,000 proteins, 3 million proteins, whatever it is, for 2 different keywords. So you can play around with this later all you want. It's a function. All you have to do is change your keywords here. So we're going to say we want antibodies. In this example, we're just going to say we want antibodies, and then we want things that aren't antibodies. And we want 2,000 of them. That's going to be our data. So we can run these cells, press the little button, or hit Shift Enter. It's going to go get the data, and it's going to pull the data, antibody proteins as X and not antibodies as Y. Alright, so now we can check and make sure how many proteins did we get for each class. 2,000 each, that's good. And let's just look at the first antibody protein. So we're going to say antibody protein, that's X. We're going to look at the first one, and it looks like this string of one-letter amino acid codes. That's great. That's exactly what we want, right? We can look at the next one. Okay, so the next one, it's a if I could type properly. It's a different one, right? It's a different protein. You can tell the length and the composition is totally different, but it's still in the same format. So, like I said, we turn these into number vectors. So we have a little function here that's just going to say, hey, each letter, assign that a unique number, and then we're going to change the letters into numbers. Very simple. So we're going to do that for all the proteins, and you end up with something like this here in cell 11, right? So you can see M is 12, whatever 3 is, 18, that's this thing down here. Okay. Now we have number. Now we've gotten our data. Step 1, done. Step 2, format it, done. We need to clean it up and make sure it's ready to be used. That's what we're going to do now. So let's find the lengths of all the proteins. We're going to do that, and then let's find the biggest ones, the biggest protein lengths, and then let's get the max, min, and average for both groups, antibodies and not antibodies, and that's down here. So the biggest one is 5,654, and the smallest ones are 5 for the antibodies, but the average is much less, right? So the average is way less than the max. So let's go ahead and look at what these protein lengths look like. So if you just took every protein and you plotted its length, every antibody, that's what you get in this plot here. So you're saying, okay, just on the x-axis is just protein number 1, protein number 2, antibody number 3, antibody number 4, and how long it is, how many amino acid residues it consists of. We're going to do the same thing for y. And we can already see that there's a lot of outliers, right? Like there's these really big ones. We don't need that. We have 2,000 proteins. We don't need those really big ones. It's just going to make things complicated later. So we're just going to take out everything that's less than 100 and greater than 1,000 in both the antibodies and not antibodies. And then we've printed out how many we have left. Okay, so we have 1,677 and 1,743. That's pretty balanced. There's no need to have to balance that. It's almost the same amount. So that's fine. We can just keep moving right along. So now let's look at the lengths again, and this time we're going to use a histogram plot. So we're just saying, this is the plot I showed in the slides. We're saying for things that are about this length, it's probably like 100, 150. There's about 500 of them in the antibody class and about 300 of them in the non-antibody class, and etc. You can extrapolate. So this is actually a pretty good indicator, right? So if you were going to build a model, if that model was just going to predict which class, if it's X or Y, if it's antibody or not antibody, just based on sequence length alone, it probably wouldn't do very well. It'd probably get about 50%, because 50% of the time, antibodies are longer than non-antibodies, right? I mean, you could do the actual math and see what exact percent, but you wouldn't do very good just guessing off length, which is what we want. We want to build a model that is actually looking at the composition and not just the length or some arbitrary feature. Because if we were doing that, we could just do that by hand. We wouldn't need a computational model. Okay, so we got that. Now we want to look at the actual composition and say which residues start the sequences and end the sequences. So we got a little function for that. We're going to do it for both antibodies and not antibodies. Now we're going to plot just the starting ones. And you can see that in both groups, it's almost unanimous that all proteins start with M, which is what we would expect from our basic knowledge of biology, right? Okay, moving on. Now we can do the same thing for the ending, which is going to be a lot more variable, as we can see here. So if you ever want to comment out a line, you can just hashtag it and it'll show you this stuff. So anyways, you could see how this technique would be useful, right? There's a little bit of a glitch in there. We're not going to worry about it. But you could see how this kind of investigation really helps you get to know your data, and you can understand what kind of features, how complex your model needs to be. Okay, that's great. That's wonderful. Let's talk about how to make these one-hot images. We have a function for that. We're going to turn our X proteins and our Y proteins into images, and we're going to look at the very first one. This is what it looks like. It's just a bunch of dots, right? So remember, the Y-axis is protein position, and the X-axis is going to be which letter it is. So A is going to be 0, B is going to be 1, C is going to be 2, so forth. You could do this one if you're not really understanding this. Try to do this by hand. Try to just say I have a protein A, B, C, D, and for each position I'm gonna mark the respective column. It's kind of like filling out a Scantron, right? When you bubble in your name, exact same thing. Okay, so we got our images. Now we're done with all that stuff. That's great. Now we can do principal component analysis, logistic regression, convolutional neural network, and then we'll interpret our results, and we're all done. Okay, so with PCA, first thing we need to do is we need to pad all of our proteins to be the exact same length, because it's going to take in each position as a feature. Each of those features represents a dimension. So we're going to pad everything to 1,000 because our biggest protein is no bigger than 999. So we're going to have one extra space on every protein at the end, and you'll see why later. But every protein gets padded to 1,000. And then we're going to put both X and Y together. And we're going to use this thing called a data frame to handle the data, to keep it organized. This is what that looks like here in cell 35. You can see on the Y, what would be the Y-axis here, or the rows, are going to be each of these is a protein. And then on the columns, that is the position. And I padded everything with the number 23 because the number 23 isn't used in our FASTA sequence, because we encoded the alphabet as numbers, like I was saying earlier. So it's just a way to encode without having to use zeros. It's generally a better practice for models. So you can see most of these aren't that long. That's why they're getting 23s towards the end. Okay, so now what we want to do is we want to say, we're going to change the color, whether or not it is a antibody or a not antibody protein. So we do that. Now we're actually doing the PCA. If you notice here in this cell right here, the actual PCA is only 2 lines. So we've already spent 36 cells just getting our data organized, which is very important and tedious. And then the actual PCA model is only 2 lines. You're going to see that a lot. Taking time to organize your data is normally the biggest and hardest step, and the models are very easy. So we run that, and we see that if we have 2 principal components, component 1 has the majority of the decision-making process, and component 2 not so much. So we can plot these results based on our data, and you get this beautiful plot here. 0 is going to be, just by the way we colored it earlier, 0 is going to be non-antibodies, and orange 1 is going to be antibodies. So you can tell that even though our data was very high dimension, there's not an easy way to reduce this and figure out the most separable case, right? You can't look at this plot and say, oh, I could easily predict where an orange dot or a blue dot is going to go. And each of these dots represents a protein, right? Based on 2 dimensions, which is some combination of the columns up here. Okay, so hopefully that makes sense. There's a lot of stuff online about PCA if you're it's not too complicated to understand, but you can see it's easy to do. Alright, so now in order for us to do logistic regression, because PCA wasn't good enough for us, we need to format our data into our training and validation, right? We're not going to use testing for logistic regression because we're not going to configure the model, so we're just going to use training and validation right now. And we need to create labels. So just like in the PCA, how we colored each of these things, each of these dots as, you know, are you an antibody or not? We need to do that down here in our actual data set. So remember how I said earlier that we left a spot at the end when we padded? In that spot, we're going to say, are you a 0 or are you a 1? Are you an antibody or are you a not antibody? So we're going to do that, and then we're going to select out random choices for our validation set, and we're going to select those proteins from the training set, and then we're going to say, okay, the training set is now everything that wasn't selected for validation, and we're going to put the X's and Y's together for both the trained data and the val data. We're going to shuffle them up. It's very important because maybe you read in your proteins in alphabetical order or something like that. You need to shuffle your data so your model can get a robust interpretation of what's going on. And if we look at the amount of data, we have roughly 2,395 altogether for training and around 1,000 altogether for validation. And we can say that our labels are the last column in all the data, right? So for each protein, the last column is going to be the label. And we can check our shapes and say, yep, we have the same amount of labels as data. Okay, now we are. I'm going to show you how easy this is. You're probably like, oh man, how are we going to get through all this stuff? We're running out of time. Easy. I told you, the data is the hardest part. So you just say our model is going to be this logistic regression, LR, that we're using from scikit-learn. It's a organization, a library for Python. We're coding in Python. And we've created our model. And then we've fit our model. You can see up here, we've fit our model to the training data and the training labels. So the model's already created and it's already been trained, so to speak, or fit to the data that we gave it, which was the training data. And now we are going to do the prediction on the validation data set. So you say model.predict val data, and it spits out all of its predictions. So for the first 3 proteins, it predicted that these guys are not antibodies. And if we look at the true labels, right, it got those 3 correct. However, for the last 3, it got one of them wrong. It said that it shouldn't have been an antibody when it really was. So we can get an overall score of the model. It's about 64% accurate. It's not very good for us. It's statistically significant because it's a binary question, but still 64, I think we can do better than that. So let's try a convolutional neural network. Now, this might get a little hairy with all the code. Don't worry about that. Just try to get the overall process. Again, the first thing we're going to do is define our model. We have convolutional layers, dropout, ReLU, things like that. That's our model. And then we're going to use the GPU. Remember I said we need to change the runtime type to use GPU accelerator. Now we have to say in the code, use the GPU. It should print out CUDA. If it doesn't, that's okay. Sometimes Collaboratory doesn't let you use GPUs based on how many people are accessing their servers. But so here we've defined what the model is going to look like, and now we want to call the model and put it to the device, which is the GPU. And this particular model configuration, later you can play around with changing the filter size and stuff like that and see how well it does. This one has 131,000 trainable parameters. So that is basically the dimension of the loss gradient. That's huge. A human could not do all these advanced calculus computations quickly, and you'll see how quick this is. We're going to use cross entropy loss, and we're going to use standard gradient descent to update these trainable parameters. That's how we're going to learn. That's how our model is going to learn these feature maps. These are just mathematical functions that say, hey, when you get this weight and you get this data, do this math and then update accordingly. It's just packaged into a really nice one line of code. So now we're actually going to run the model, and when we run the model, we're going to take out little tiny batches. So we're not going to run everything all at once. We're going to say, okay, look at 8 proteins at a time, make your decisions, and then update yourself, and then do that whole process for all the data based on however many epochs, right? So your epochs, one epoch, you look at all the data based on your little tiny batches, and then you update your weights and stuff like that. So we're going to define that, and then we're going to train the model with the training data. Right? We're only going to do it for a few epochs to save time, and we're going to do it with a small batch size of 8. So you can see the model is going to start training. And there it goes. The loss starts at 0.7. It's not very accurate. It's getting near chance. On the second time around, it's looked at all the proteins and it's updated itself. It got a little better. The loss got lower. The accuracy got higher. It keeps getting better. This is what you want to see. We're already up to 82% accuracy, right? So that's pretty good. I know it says point. That's just because I didn't multiply the thing properly. Just go ahead and multiply by 100 there. So in 5 epochs, we got up to 98% accuracy. That's pretty good. That's a lot better than our logistic regression, and we have a pretty small model considering some other models you've probably heard about, ResNet, AlexNet, can be millions and billions of parameters. Ours is only 100,000. All right, so we trained with the training data. Now we want to test the validation data. So we're not going to update any of our parameters. We're just going to use what we learned to see how well we do on other data. And we do really good. We get 99% accuracy. And yep, so we're doing good, right? So here you may want to say, okay, I want to do even better. Then I'm going to change my batch size, and then you would retrain, and then you'd revalidate. And we're leaving off the last 500 of the validation set. We're going to use that for testing. So that's it. That's the CNN. We're done with all of our models. We have a good model. Now we need to interpret it. We're going to do an area under the curve ROC plot. We're going to use the probabilities that the model predicted and the actual predictions to generate false positive rates, true positive rates, and get our AUC, which is 0.16. And when we plot that, this is our ROC curve. So there's lots of ways to interpret this. I'll leave that up to you guys later on. This is just more on how do you generate these things that people are going to want to see and that you're going to want to see to interpret your results and report your model accordingly. So now we can look at sensitivity, specificity, and recall. We can make a confusion matrix that says, okay, when we were supposed to predict non-binding, that's actual 0, how many times did we get that correct? So a lighter color means we did better, and you can see we predicted like 260 proteins correctly. and we don't predict any incorrectly. So that would be actual 1 predicted 0. You can kind of see how this is set up. So from that, we can get these precision metrics we wanted. F-score is kind of just like a ratio of your false positive, your precision, your recall, and your specificity. It's just a way to have one number to evaluate your model. Let's say you're changing or comparing models. Sometimes you just want to have one number to look at instead of 3 of them. So that's that. Last thing we're going to talk about is feature map visualization. So we're going to use these one-hot images, and we're going to say, let's just see one image, right? Let's just look at one of our one-hots. If you go over here to the side, you can look at files, and you can open the picture, you can download it, and it'll get bigger. Okay, so this is what we're expecting. We're expecting a lot of positions and not a lot of letters choices. So we're going to look at the first convolutional layer, and we get this feature map, right? Now we can look at this second convolutional layer, and we get another feature map. So let's go ahead and look at more closely at that second feature map, because I know we can't really see it. You just go over to here to the side, and we're going to download this, and we're going to open it, and hopefully you guys can see it. But basically, if you look closely at it, you should see activations for certain positions that are important, and it's also telling you in this printout, the least activation was 0, the most activation was 5, and if you print out the prediction, it'll tell you, does it think this is an antibody or not. So this doesn't make a whole lot of sense to us right now because we're looking at one-hot images of proteins, but let's say that you were looking at pictures that you got from a microscope of X-ray crystallography or something like that of proteins. Then you may actually really be interested in these feature maps because it may show areas of interest for binding or for whatnot, however you're structuring your problem. So that's it, guys. I hope you don't have too many questions. If you really have a question, you can email me, email Dr. Pavlovic. She'll send it over to me. I'll try to answer your questions. Please feel free to play around with this code in your own copied version. It's okay if you don't know a lot of code. Just try changing little things and see what happens, right? Then you can start to get a feel for, okay, a little bit here and a little bit there. Does that make the model better or worse? Stuff like that. One thing you could try to think of a fun problem to tackle with Uniprot and change the keywords. So the keywords up here in the beginning, you can just change these out. Instead of antibodies and not antibodies, you could do whatever other keywords you want. You can go to Uniprot and look at their list of keywords. Alright, everybody, so I hope that was helpful and a little bit insightful for the overall methods you would use and what it actually takes to do this kind of computational analysis. It's not too hard. I'm sure everyone would get the hang of it the more you read and get into it, and I hope everything made sense. Hopefully Dr. Han will explain a little bit more how these basic techniques get extrapolated to really advanced stuff. So thanks for listening, guys.